Hi, welcome to Dr. Komal Lectures. As in the previous lecture, we have studied about the virus and its properties. In case you haven't seen the video, you can check the link given in the description box below. In today's class of microbiology, we are going to study about the coronavirus, its properties, its classification, various types, its genome, and its structure. So let's begin. What is the coronavirus? Today, we all have heard about coronavirus and coronavirus disease, that is COVID-19. In today's lecture, we will study about various types of coronaviruses, and I will discuss about the genome and structure of novel coronavirus, which is SARS coronavirus 2. Okay. A group of spherical enveloped RNA virus which carry petal-shaped papillomers. Now, papillomers are the glycoproteins which are on the surface of the virus. This group has been classified as coronaviruses. Many serotypes of human coronaviruses have been recognized. They appear to be the second most commonest cause of the common cold. And which particularly affects in winter season next to the rhinoviruses. This is the mild infection, but the lethal varieties can cause severe acute respiratory syndrome, that is SARS, Middle East respiratory syndrome MERS, and the COVID-19, that is the coronavirus disease. Let us study about the discovery of the virus. Coronaviruses were first discovered in 1930s, and the human coronaviruses were discovered in 1960. Earliest cases were from the patients with the common cold. Many other variants have also been identified, including the severe acute respiratory syndrome coronavirus in 2003, Middle East respiratory syndrome coronavirus in 2012, and SARS coronavirus 2 in 2019. Nomenclature of the virus. So, this is coronavirus. Why is it called as coronavirus? Let us see. It has been derived from the Latin word corona, which means crown. Now, this refers to the appearance, which is a basic characteristic appearance of the virions, which have large projections or spikes, creating an image of the crown. So, corona. Now we will study about MERS. This will be a brief introduction about MERS and SARS. MERS is Middle East respiratory syndrome. MERS was first seen, or its outbreak was in 2012. It is also called as camel flu. So, coming to its definition, it is a viral respiratory infection which is caused by Middle East respiratory syndrome coronavirus. Now, this virus is a beta coronavirus which is derived from the bats. And camels are believed to be involved in its spread, thus also known as camel flu. Coming to the symptoms of MERS, the symptoms range from mild to the severe form, such as there can be fever, cough, diarrhea, shortness of breath, etc. Now, mortality of Middle East respiratory syndrome is 1/3 of the diagnosed cases. MERS was spread in Middle East, as the name suggests, and South Korea, Saudi Arabia. Coming to SARS. SARS is severe acute respiratory syndrome. SARS is caused by severe acute respiratory syndrome coronavirus. The outbreak occurred in 2003 and it infects humans, bats and civets. Coming to the symptoms of SARS, there is muscle pain, fever, headache, cough, dyspnea and pneumonia. Also, other findings in SARS patient, there is decreased value of lymphocytes in the patient's blood. SARS was seen in more than 29 countries and about 8,000 cases were noted. Now let us understand what is novel coronavirus or severe acute respiratory syndrome coronavirus 2, the recent famous coronavirus, which causes COVID-19, that is coronavirus disease. Now, SARS coronavirus 2 is positive sense single-stranded RNA virus. WHO has designated this pandemic as public health emergency of international concern. It has globally affected all the nations including Italy, USA, India and many other nations. It was discovered in Wuhan, China, and the outbreak was in December 2019. Classification. On February 11, International Committee on Taxonomy of Virus have identified novel coronavirus as severe acute respiratory syndrome coronavirus 2. So, the reme is Riboviria, phylum is Incertaceidis, order is Nidoviralis, and the family is Coronaviridae. Genus is Beta coronavirus. Subgenus is Sarbecovirus. Species is severe acute respiratory syndrome related coronavirus. And the strain is severe acute respiratory syndrome coronavirus 2. Origin of SARS coronavirus 2. In December 2019, in Wuhan city of China, in its meat market, the first outbreak of coronavirus disease has occurred. It is believed to be of zoonotic origin, animal origin, suggesting it emerged from bat-borne virus. Also, interestingly, an intermediate animal reservoir, that is pangolin, is also thought to be involved in its introduction to the humans. Coming to the virology, human to human transmission of SARS coronavirus 2 have been confirmed. In my previous class, I have mentioned about various modes of transmission of viruses. Since COVID-19 is a droplet infection, thus the transmission occurs by respiratory droplets, that is from coughs and sneezes of the infected person within a range of 6 feet. Also, indirect contact with contaminated objects or surfaces, which are known as fomites, can also spread the infection. Now, human coronavirus can remain infectious from 2 hours to 9 days. Let us study about the viability of this virus. At temperatures around 4 degrees Celsius or 39.2 degree Fahrenheit, coronavirus remains viable up to 28 days. And on plastic or steel surfaces, it can remain viable for 72 hours, that is 3 days, on cardboard for 24 hours, and on copper for about 4 hours. Inactivation of the virus. It gets inactivated by contact with soap, as the soap destabilizes the lipid layer. The outer envelope contains lipids. Let us study about the structure of the coronavirus, that is novel coronavirus or SARS coronavirus 2. Each SARS coronavirus 2 virion is around 50 to 200 nanometers in diameter. The novel coronavirus is, according to Baltimore classification, positive sense single-stranded RNA virus. It belongs to class 4 of Baltimore classification. Now, it has 5 structural proteins, namely spike, envelope, membrane, nucleocapsid, heme agglutinin esterase dimer, and an RNA genome. SEM protein together creates the envelope. Let us see this using the example of diagram of coronavirus. So, this is a spherical structure of the coronavirus. These are the petal shaped papillomers. These are the glycoproteins which will help in the attachment to the host cell. This is the E protein. This one is the membrane protein, M protein. Now I am making heme agglutinin esterase dimer, that is HE. This is the viral RNA genome and N protein. Let us label it. This is spike glycoprotein S, E protein, membrane protein, RNA genome and N protein. This is heme agglutinin esterase dimer HE. This is the outer lipid layer envelope. SARS coronavirus 2 has affinity to the receptor angiotensin converting enzyme ACE2 on human cells to use them in the mechanism of cell entry by the process of endocytosis. The spike proteins on the viron attaches to ACE2 receptors. They may also use biosigin for the process to enter inside the cell. Angiotensin converting enzyme 2 is expressed by epithelial cells of intestines, kidneys, blood vessels, and most abundantly in type 2 alveolar cells of lungs. Thus, lungs are affected mostly in COVID-19 disease. Let us study the genome of SARS coronavirus 2. Genome means genetic material of an organism. Coronavirus 2 contain positive sense single-stranded RNA genome, the size of which varies approximately between 26.4 to 31.7 kilobases. The genome has a 5' methylated cap and a 3' polyadenylated tail. So, let us make the diagram or the structure of the genome of SARS coronavirus 2. These are the various polyproteins. This is 1A, 1B, S protein. Various other accessory proteins envelop, membrane and nucleocapsid. SARS coronavirus 2, the size varies 29.7 kb. This is 5' leader untranslated region. These are open reading frames. Open reading frame 1A and 1B encode the replicase or transcriptase polyproteins which self cleaves into non-structural proteins. And the later reading frames encode 4 major structural proteins, namely spike proteins, membrane proteins, envelope and the nucleocapsid. This is spike protein, envelope, membrane protein and the nucleocapsid. This is the cap, 5' cap, and this is the 3' tail. Life cycle of severe acute respiratory syndrome coronavirus. 2. This is a cell. This is the virus. These are the spike proteins. Now, the infection begins when the viral spike glycoprotein S attaches to the angiotensin converting enzyme 2 receptor on the host cell. First process is the attachment process. Now, the protease of host cell cleaves and activates receptor attached to the spike proteins and allows the viral entry through the process of endocytosis. On entry, there is uncoating and the viral genome enters the cell cytoplasm. Now, the coronavirus RNA attaches to the ribosome of the host cell for translation. Translation is a process by which viral mRNA is translated into viral proteins using host cell machinery. So, the translation of open reading frame 1A and 1B occurs, and then the replication and translation of the further other frames occur. Host ribosome translates virus genome and form long polyproteins. The polyprotein has its own proteases which cleave the polyprotein into multiple non-structural proteins. Now, the number of non-structural proteins coalesce to form a multiple protein replicase transcriptase complex. And ultimately, the synthesis of positive sense mRNAs occur in the ribosome. Various proteins are being formed. Thus, protein synthesis have occurred. Viral protein and genome RNA are assembled into virons in endoplasmic reticulum, Golgi apparatus, and then transported via vesicles and released out of the cell. Now, the mRNAs are formed and they are translated by host ribosome into structural and accessory proteins. RNA translation occurs in the endoplasmic reticulum. RNA translation occurs in endoplasmic reticulum. Now, the viral structures that are the spike proteins, envelope, membrane, they make their pathway towards Golgi apparatus after being assembled in the endoplasmic reticulum. The red portion I am showing is the nucleocapsid, yellow protein is the envelope protein, whereas the green is the membrane protein, and the protein shown in black is the spike proteins, which will further help in the attachment process. This is the Golgi apparatus. And now the newly formed virons are assembled in the vesicles. The M proteins are required for assembly of virus following its binding to nucleocapsid, and thus the newly formed viruses are released from the host cell by the process of exocytosis. So, here we have understood the whole life cycle of the SARS coronavirus 2. How does it enter the cell and how does it replicates to form the multiple viruses which are released. So, this was the structure and the life cycle of coronavirus. In our next lecture, we will study about the disease caused by coronavirus, that is COVID-19 or coronavirus disease, its signs and symptoms, spread, prevention, how does it harm the human body, and the various FAQs regarding it. Thanks for watching. Subscribe my channel for further videos. Hit the like button and share it among your friends.